Please subscribe and hit the like button. Hello everyone, my name is Jong Hyun Yu. I will present about electromicrobiology, specifically focusing on the ecophysiology of phylogenetically diverse electroactive microorganisms. The field of electromicrobiology originated from foundational studies on microbial fuel cells, MFCs, systems in which electroactive microorganisms generate electrical current by transferring electrons to an external electrode as part of their respiratory process. These early investigations provided the first tangible evidence that certain microbes could directly interact with solid-state electron acceptors, laying the groundwork for a new understanding of microbial bioelectrochemical activity. Initially regarded as a phenomenon limited to engineered systems such as bioelectrochemical reactors, this electron transfer capability is now recognized as a widespread and ecologically significant trait among diverse microbial taxa. Recent advances have demonstrated that microorganisms capable of extracellular electron transfer are not confined to laboratory conditions or synthetic environments. Instead, they are found across a wide array of natural settings, including marine and freshwater sediments, anoxic soils, hydrothermal vents, subsurface rock strata, and even within host-associated microbiomes. In these environments, electroactive microorganisms participate in natural redox processes by coupling the oxidation of organic or inorganic substrates to the reduction of insoluble electron acceptors such as iron, manganese, or sulfur compounds. Their metabolic activities contribute to critical biogeochemical cycles, influence mineral transformations, and mediate interspecies electron transfer within complex microbial communities. Thus, what began as a narrow investigation into microbial electricity generation has evolved into a broad scientific domain that explores how microorganisms interact electrically with their environment, both in natural ecosystems and in engineered applications. This paradigm shift has significantly expanded the relevance of electromicrobiology beyond power generation, highlighting its importance in environmental science, geochemistry, and synthetic biology. In this presentation, I'll focus on what electromicrobiology is, the diverse groups of electroactive microorganisms, their roles in various natural ecosystems, detailed mechanisms of extracellular electron transfer, and emerging practical applications and future perspectives. Electromicrobiology is an interdisciplinary scientific field dedicated to studying microorganisms capable of exchanging electrons with their external environment through a process known as extracellular electron transfer. The roots of electromicrobiology trace back to early investigations of microbial fuel cells, where researchers observed microbes transferring electrons directly to electrodes to produce electrical current. However, the field evolved. It became clear that these electroactive microorganisms exist in numerous natural environments and significantly impact ecological processes and biogeochemical cycles. In natural ecosystems, microorganisms rarely live isolated. Instead, they frequently interact electrically with minerals, organic molecules, and even other microorganisms. This electrical interaction is primarily achieved through unlike conventional microbial respiration, where microorganisms transfer electrons internally to soluble acceptors that freely diffuse into the cells, electroactive microbes must interact external with insoluble acceptors like Fe3 oxides, manganese oxides, electrodes, or other microbes. These extracellular electron acceptors or donors typically cannot pass through microbial membranes due to their size, charge, or insolubility. Hence, microbes have evolved sophisticated biochemical pathways and structural adaptations specifically tailored for extracellular electron transfer. One classic example of naturally occurring electromicrobial activity is the microbial reduction of Fe3 and Mn4 oxides in anaerobic soils and sediments. Fe3 oxide reduction, in particular, is historically and ecologically significant. It is believed to be among the earliest forms of microbial respiration on Earth, evolving billions of years ago. Microorganisms like Geobacter and Truyonella effectively transfer electrons extracellularly to these insoluble minerals, altering the geochemistry of their habitats and playing critical roles in carbon cycling, contaminant remediation, and mineral transformations. Electroactive microorganisms are incredibly diverse in terms of their phylogeny, metabolic capacities, and ecological adaptations. These microorganisms are not limited to a single taxonomic group but are distributed across a broad range of bacterial and archaeal lineages, illustrating the evolutionary versatility and widespread ecological importance of extracellular electron transfer. These electroactive microbes are further classified based on their metabolic processes, Respiratory electrogens use external electron acceptors in their respiratory electron transport chains to generate via oxidative phosphorylation. For example, Geobacter sulfurgesins and Schuyinella-1-adensis oxidize organic substrates completely to carbon dioxide, transferring electrons externally to Fe3 minerals or electrodes through cytochrome-based electron transport systems embedded in their membranes. Fermentative electrogens, on the other hand, produce primarily through substrate-level phosphorylation during fermentation. Electron transfer to external electron acceptors is typically not their primary energy-generating pathway but serves as an alternative electron disposal strategy. Examples include gram-positive bacteria such as Listeria monocytogenes and Enterococcus faecalis, which transfer electrons to soluble extracellular electron acceptors like flavins and humic substances through flavoprotein-based mechanisms. In addition to bacteria, several archaea exhibit electroactivity, particularly methanogenic archaea involved in direct interspecies electron transfer. For instance, methanogenic archaea such as Methanosarcina and Methanotrix directly accept electrons from electroactive bacteria, facilitating efficient and stable methane production in anaerobic digesters. These archaea often utilize cytochrome-based electron transport pathways similar to their bacterial counterparts. A unique and fascinating group of electroactive bacteria, known as cablobacteria, form centimeter-long filaments composed of thousands of cells. These filaments conduct electrons across sediment layers, linking anaerobic zones, where sulfide oxidation occurs, to aerobic zones, where oxygen reduction takes place. Cablobacteria thus dramatically impact sediment chemistry, affecting the cycling of iron, sulfur, phosphorus, and trace metals. Let's take a closer look at Figure 2, which visually illustrates the diverse functional roles electroactive microorganisms play in different electromicrobiomes. This figure is organized into distinct environmental contexts where electroactive microbes significantly impact the ecosystem. In anaerobic soils and sediments, electroactive microorganisms play key roles in electron transport across different redox zones. In upper layers, cablobacteria, which form multicellular conductive filaments, transfer electrons from anaerobic sulfide oxidation zones up to aerobic layers, where oxygen is reduced. This long-distance electron conduction significantly alters sediment geochemistry, affecting iron, sulfur, phosphate, and metal cycling. Within deeper anaerobic layers, respiratory electroactive bacteria, REBs such as Geobacter, Desulfuromonas, and Rhodophrax directly reduce insoluble electron acceptors like Fe3 oxides. This process releases dissolved Fe2 and trace metals, affecting groundwater chemistry and contaminant mobility. G. Arsenic release. Fermentative electroactive bacteria, FEB, including Clostridium and Listeria, ferment organic substrates, donating electrons extracellularly or to respiratory bacteria via direct interspecies electron transfer, DIE. Anaerobic methane oxidation is mediated by electroactive archaea such as methanopridines, which oxidize methane while transferring electrons to Fe3 or sulfate reducers, controlling methane emissions in sediments. In geothermal environments such as deep-sea hydrothermal vents, electroactive hydrothermophiles, including Pheroglobus and Pyrobaculum, utilize Fe3 as electron acceptors to oxidize hydrogen and organic compounds, forming unique electroactive microbial communities adapted to extreme temperatures. These processes are critical to mineral cycling and microbial survival under extreme conditions. In engineered systems like anaerobic digesters, electroactive microorganisms significantly influence bioenergy production. Fermentative bacteria degrade complex organic waste into simpler organic acids and alcohols. These products are further oxidized by electroactive syntrophic bacteria, transferring electrons directly to electron-accepting methanogenic archaea, enhancing methane production efficiency and digester stability. In animal intestines, electroactive microorganisms influence gut ecology and health. Fermentative electroactive bacteria, including Enterococcus and Listeria, perform extracellular electron transfer using soluble electron shuttles, interacting with other microbes and possibly affecting oxygen gradients near the gut mucosal lining. These electroactive interactions potentially contribute to maintaining intestinal homeostasis, modulation, and microbial community structure, underscoring the importance of electroactivity in host-microbe relationships. On metallic surfaces, electroactive microorganisms significantly impact corrosion processes. Electroactive bacteria such as Desulfovibrio and Geobacter species facilitate anaerobic corrosion by extracting electrons directly from iron-containing metals. thereby accelerating deterioration of metal structures such as pipelines and storage tanks. Understanding these microbial corrosion mechanisms informs innovative corrosion control strategies, including biological methods like pre-colonizing surfaces with protective, biomineralizing microorganisms. Microbes evolved distinct mechanisms to transfer electrons extracellularly. These mechanisms show how electroactive microorganisms adaptively evolved complex electron transport systems for survival and growth. Gram-negative bacteria like Schwanella and Geobacter use porine cytochrome complexes. These protein complexes bridge electrons across the outer membrane through multi-heme C-type cytochromes, efficiently transferring electrons to external electron acceptors. Gram-positive bacteria such as Listeria and Enterococcus employ flavoproteins embedded in cell walls to transfer electrons externally to soluble electron acceptors like flavins and humic substances. Such mechanisms bypass structural limitations of thick gram-positive cell walls. Some electroactive microbes secrete redox-active molecules called electron shuttles, like flavins, phenazines, and humic substances. These shuttles diffuse and transport electrons to distant solid phase electron acceptors, enhancing microbial electron transfer efficiency. Certain microorganisms, notably Geobacter, produce protein-based conductive nanowires called ecophyli. Ecophyli extend electron transport capabilities beyond cellular boundaries, facilitating direct interactions with solid phase minerals and interspecies electron exchange. Cablobacteria, filamentous bacteria spanning millimeter to centimeter distances, electrically couple anaerobic and aerobic zones within sediments using specialized internal conductive fibers, drastically influencing sediment biogeochemistry. It is a diagram illustrating that cells are arranged side by side like wires, and there is a structure that carries electrons inside each cell. Experimentally, this structure was observed in the Cablobacteria electrothix and Electroma. Electromicrobiology has inspired multiple innovative applications. Environmental remediation, stimulating electroactive bacteria. For example, adding electron shuttles or conductive materials enhances biodegradation of persistent contaminants such as petroleum, hydrocarbons, or immobilization of radioactive uranium in groundwater. Bioenergy production, optimizing diet processes through addition of conductive materials like biochar or activated carbon significantly increases methane yield in anaerobic digesters, offering robust, economically viable renewable energy solutions. Corrosion mitigation. Eco-friendly corrosion management strategies emerge from electromicrobiology research, including biofilm formation by beneficial microbes that protect metal surfaces by preventing colonization of harmful electroactive microbes. Electron biomaterials development. Novel biomaterials, such as conductive protein nanowires harvested from electroactive microorganisms, are emerging. These nanowires are promising components for sustainable electronics, sensors, and energy harvesting devices. Future research in electromicrobiology aims to clarify unknown mechanisms of electron transport, microbial interactions, and genetic regulation of pathways. Knowledge will facilitate biotechnological advances in energy, environmental sustainability, and bioelectronics. Electromicrobiology bridges microbiology, ecology, and engineering, revealing extraordinary microbial capabilities to transfer electrons across cellular boundaries. Understanding these electroactive microorganisms is reshaping environmental technologies, renewable energy systems, and sustainable material development. The future potential of this interdisciplinary field is immense, and continuous exploration promises transformative impacts for society and the environment. Thank you very much for your attention.